function of cell division is for cells to grow and replace damaged cells. Asexual reproduction is the production of offspring that are genetically identical to the parent cell. Sexual reproduction is the production of offspring that are different from the parents, and two parents contribute half of the genetic information to the offspring. Interphase is the non-dividing phase of a cell's life, and it consists of about 90% of the cell's life. During interphase, the genetic information, DNA, is in its uncondensed form, chromatin, in the nucleus. The nucleus is surrounded by the nuclear envelope. During interphase, cells carry out normal processes, such as growth and DNA replication in preparation for cell division, as well as double-checking the replicated DNA for mistakes. Mitosis is asexual reproduction at the cellular level that increases cell size and replaces damaged cells. The stages of cell division are as follows. In prophase, the nuclear envelope breaks up. The chromatin condenses into visible chromosomes. Each chromosome consists of two identical sister chromatids joined at the center by a centromere. Centrioles, found only in animal cells, produce microtubule spindle fibers, which attach to structures known as kinetochores on the centromeres of chromosomes. There are two kinetochores per centromere, one for each sister chromatid. In metaphase, the spindle fibers coordinate the movement of the chromosomes to the center of the cell, which is known as the metaphase plate. Spindle fibers movement occurs using motor proteins known as actin and myosin. In anaphase, centromeres between sister chromatids split, and the spindle fibers pull identical sister chromatids to opposite ends of the cell using their motor proteins. Spindle fibers that are now not connected to chromatids lengthen the cell. In telophase, chromatids, now known as chromosomes, arrive at opposite poles, and two new nuclear membranes form. In cytokinesis, the cytoplasm divides to form two new daughter cells. In animal cells, a cleavage furrow forms, and the cytoplasm pinches in two. In plant cells, vesicles from the Golgi bodies bring cell wall material to the middle of the cell, and a new cell wall splits the cytoplasm. Meiosis is cell division that forms haploid gametes from diploid cells in preparation for sexual reproduction. Because each gamete is haploid, each parent can contribute half of the offspring's genetic information in the form of one set of homologous chromosomes. Homologous chromosomes are structurally identical chromosomes that may contain different forms of genes, known as alleles. Meiosis consists of two cell divisions, although DNA is only replicated once before the first division. After DNA replication, each chromosome consists of two sister chromatids joined by a centromere, just like prior to mitosis. In prophase 1, chromatin condenses to form visible chromosomes, and the nuclear envelope breaks down. Synapsis is the formation of tetrads. Replicated sister chromatids pair with their replicated homologous chromosomes. Each tetrad consists of two chromosomes, or four sister chromatids. Crossing over is when homologous sister chromatids intertwine and swap pieces to increase genetic variability. Spindle fibers form from centrioles and attach to the centromeres. In metaphase 1, the tetrads, which are joined at specific points called chiasmata, are moved to the middle of the cell by the spindle fibers. In anaphase 1, the tetrads split and homologous chromosomes are pulled to opposite ends of the cell by spindle fibers. Centromeres remain attached between sister chromatids. Unattached spindle fibers elongate the cell. In telophase 1, the nuclear envelope forms around chromosomes as they become uncondensed. Cytokinesis occurs and two daughter cells are formed. Meiosis 2. In prophase 2, the chromatin condenses into visible chromosomes and the nuclear envelope breaks down. Spindle fibers form and attach to kinetochores of the chromosomes. In metaphase 2, the spindle fibers pull the chromosomes to the metaphase plate in the middle of the cell. In anaphase 2, centromeres between sister chromatids split, and spindle fibers pull sister chromatids to opposite ends of the cell. Unattached spindle fibers elongate the cell. In telophase 2, nuclear envelopes form around the sister chromatids, which are now referred to as chromosomes, and the genetic material uncondenses. Each cell now has only 23 chromosomes, meaning that it is haploid. Cytokinesis occurs and there are 4 daughter cells. Differences between mitosis and meiosis. In mitosis, there's asexual reproduction. Daughter cells are genetically identical to parent cells. There is no crossing over. Mitosis consists of a single cell division and produces 2 daughter cells. Meiosis is sexual reproduction. Daughter cells have half the number of chromosomes as parent cells and can be genetically different due to crossing over. Meiosis consists of 2 divisions and produces 4 daughter cells. Cancer occurs by the uncontrolled growth of abnormal cells in the body. It occurs when tumor suppressor genes, cancer checkpoints, and cell suicide genes are compromised.